he was a chemist. He did masters in chemistry, and he became a faculty in the Department of Chemistry. Prior to that, he did twice PhD, once in chemistry from PAU Ludhiana, and second one he did from Texas A&M in biochemistry. Then he came here, and he was the founding center head for Center of Biosciences. In 1980, he started this department, and after few years, I think it became Department of Biosciences, full-fledged department in 86. And then 1995 till 1995, he was a professor in this department. So he was the first head of the center and then first department head of this biosciences department in 1986. So he was retired in 1995. He was also a fellow of National Academy of Sciences, and he was Humboldt Fellow and great scientist. So we thought of, to have someone, this sort of lecture from our own department. Earlier, we started with Ramachandran Award, but Ramachandran is everybody's, many people are having that sort of lecture. But C.B. Sharma has contributed to this department. We, all of us are here because of him, and otherwise this department wouldn't have been here. Now I would like to speak about our today's speaker, Dr. Amit Sharma. There's a lot of things to read about Dr. Amit Sharma, but I would try to be as brief as possible. So Dr. Amit Sharma has done his bachelor from Purdue University, where I did postdoc, and he was the one who guided me to go to Purdue. And because I was lucky to have postdoc from his PhD supervisor, Professor Alfonso Mondragon, and from Purdue. So he said, go to Purdue, where I did my bachelor. So he was a new faculty that time in ICGP. So after doing his PhD, bachelor from Purdue, he had gone to Northwestern University and for PhD. And then afterwards, he has moved to Oxford University, UK for his postdoc, where both the places he has published Nature, Science, Cell, all top journals in PhD and postdocs. And he came here as a group leader in ICGP since 2001. And 2001, I met first time Dr. Amit that time. And later on, he became the director of National Institute of Malaria Research, New Delhi. He was there from 2019 to 22. He's the fellow of all academies, top academies in India, NASI, Indian Academy of Sciences, Indian National Science Academy, the World Academy of Sciences, National Academy of Medical Sciences, and he has all the top awards, but top awards I will mention, the Shanti Swarup Bhatnagar Award and Infosys Award. And so many more things, but I think we don't have to get into all these things because he's having very big CV. So I request, we are very proud that he's the first seminar speaker today here, and we are great. It's our great pleasure to invite you, Dr. Amit Sharma, and thank you for accepting our invitation. And it's my request that you can take the podium now and deliver the lecture. Thank you. Just before we start, I think there's one thing which I forgot. We have the senior most professor sitting, Professor R.P. Singh. Sir, I would like to request that if you can, give them bouquet to Professor Amit, sir, please. Dr. Amit, please.
Good afternoon. Thank you, Dr. Praveen, for a wonderful introduction. I apologize for a little delay in this setting up the PPT. So, it's a great privilege and honor for me to be the first lecturer in this series for Professor C.B. Sharma. I assure you that I'm not related to Professor C.B. Sharma, so you can be at rest about conflict of interest issues. Also, I want to tell you a little story. Actually, my grandfather, who was born in a village, he had an elder brother, and my grandfather decided to not be a farmer and study maths and physics. And so he was one of the first graduates from, at the time, what was called Thompson College. which subsequently in his next generation, all my uncles went to at the time it was called Roorkee University, which is now called IIT Roorkee. So I have a strong family lineage that has actually benefited from this institution. I'm really privileged to give this lecture to you. So thank you, Dr. Praveen Kumar, for thinking of me for this. I'll take you through a brief journey, but it's going to be focused on malaria. I hope you have an interest in malaria, but people who aren't interested in malaria should see it as a journey through various aspects of infectious disease science and also perhaps various aspects of structural biology. And I hope there'll be some learning lessons for everybody, despite their, if they are not specifically interested in malaria. So you can look at malaria as a proof of concept or as a thematic exercise. But I think a lot of the issues that I've touched through will be relevant to many infectious diseases. So I have always looked upon malaria as a biomarker for national development, and it will become clearer to you towards the end of the talk why I say so. Malaria has been with us for literally thousands of years. As you know, India has had malaria for a long time, but a lot of the morbidity and mortality that is usually associated with malaria has been in Africa and in Southeast Asia. But malaria is a really, really ancient disease, and I don't know if there's a pointer here. It's a really ancient disease, and um, for its spread, you need the vector, which is the mosquito, and you need the malaria parasite. And both of them are actually very old. As you know, humans sort of evolved, current species of humans, Homo sapiens sapiens, have been around on Earth for about 100,000 years. But the mosquito species has been around for some 230 million years. And the parasite species have been around for some 130 million years. So you can see that they have been here far longer than even primates have been around. And at the time, they used to infect other animals, and they continue to infect other animals. And in fact, it's a classical example of a zoonotic disease. Also, malaria was around throughout the world, essentially, except for highly, highly cold regions, which are shown in green. In 1900, malaria was rampant across all the regions here shown in red. Thank you. इसमें बोल रहा हूँ. All the regions shown in here in red. And that's really important because it's looked upon as a disease of the tropics. It's looked upon as a disease of the poor. But in fact, even in the United States, it used to be rampant in 1900s. And what we are familiar with today, what's called Centers for Disease Control in Atlanta, CDC, was actually set up as an anti-malarial unit. At the time, the malaria used to be in the swamps in Florida. So malaria was rampant throughout 1900s, and really over the past 100 years that we've been able to contain it somewhat. And what's special about malaria, it may be true for many other infectious diseases as well, is that it's marked as one of the oldest diseases that has infectious disease that has coexisted with humans. It's obviously caused most amounts of deaths and suffering worldwide and continues to do so. Humans have co-evolved with malaria, and there are genetic signatures in all us humans that are evidence that it has co-evolved with us. And so there are actually imprints of genetic diseases as is common, that actually there's been selection against infection from the malaria parasite, and humans have co-evolved to sort of avoid that, but at a certain cost. And a classical example of that, and people in this department would know, would be sickle cell anemia. In patients who are immunosuppressed for some reason or other, malaria can be cause worse outcomes. It can be congenitally transmitted from mother to child. There are, of course, 200 to 400 million cases. There is a huge problem in surveillance, so I'm not going to put an exact number to that. But 200 to 400 million cases each year, and almost half of the current Earth's population is at risk of malaria. It causes this very deaths. This is again due to surveillance, if you trust the surveillance methods currently, which are not robust and all. And if one looks at the economic cost of malaria, it's always been very high. It's about 1.5% of the GDP, and you know how even 1% of GDP manifests itself in literally huge funding for other issues. So malaria has been with us for a long time, and countering it has been a real issue for humanity on the whole. But if you're interested in sort of science and you really think that unless you get a Nobel Prize, science is not worthwhile to do, then also malaria is quite rewarding, because you can see here there have been 5 Nobel Prizes specifically for malaria over the past 100 years or so. Ronald Ross started off with, who discovered that mosquitoes spread malaria. and Charles Leverin then to show that parasites exist in our erythrocytes. Um, Julius Wagner, who it was somewhat remains controversial even today, but in First World War and later on they solved syphilis cases in patients using malaria infection because it caused high fever and improved the symptoms of syphilis. Paul Muller, of course, for discovery of DDT, and for me, most importantly and most significantly, Professor Tu Youyou, who won the Nobel Prize, the first Chinese Nobel laureate who won the Nobel Prize in 2015 for discovery of artemisinin. And it is estimated that her discovery, in fact, has literally saved close to 10 million people's lives so far since the discovery. So there have been 5 prizes so far in malaria, and you can see why that is the case, because the significance of the disease has been very tremendous for humanity. If you look at malaria over the past, say, 60 to 80 years across India, you can see that it was really rampant in 60s and 70s, and there was this huge peak, and there was a subsequent peak, but since then it has been on tremendous decline. And that really bodes well because India as a country has continuously committed itself to malaria control and spends literally thousands of crores every year in malaria control, prevention, and surveillance and treatment. And so that's why you see this very low number of cases here in 2021 of malaria. And you can imagine the caseload. And this is really the caseload in the public healthcare system. This doesn't take into account the private healthcare system, which is actually booming here. And so we don't really count those. Anyway, at the moment, the government doesn't count the private sector cases. So this is what I would estimate this to be an underestimation of the total burden. But nonetheless, it's been very encouraging for India. So you can see cases state-wise in 2020 and 2021. And these are cases greater than 20,000, for example. So it's the typical states of Chhattisgarh, Odisha, Jharkhand, which really bear the brunt of malaria, which also coincide with, to a certain extent, with poverty and forested areas and tribal cultures. And the deaths due to malaria have also come down. The deaths are less than 100 as per the national program per year. So it's really, India has done really remarkably well in bringing down the incidence of malaria over the past many decades. So how do you do surveillance of malaria? Because, India is a large country and doing surveillance for anything. And of course, everybody is now really educated about surveillance because of COVID. But surveillance of malaria is in fact far more tricky. So just as a recap for people, I really want to stress this because the title of the talk was, structural biology to epidemiology. And it's really important if you're working in infectious disease to understand epidemiology. So as you know, India is broken down into several states, and these are broken into districts, some 740 odd districts, which defines the whole of the country. Then you have CHCs and PHCs. PHCs, there are about 30,000 of them. They cater to a specific population. Districts have about 2 to 2.5 million per district, so that would account about 1,400 million people. And these are broken down into sub-centers. Now, surveillance has to start from ground up. And this is a non-trivial and an arduous process because you can imagine the paperwork required for this. So the way the national program does surveillance is, which is the national program under the Ministry of Health and Family Welfare, the national program collects data on various meliometric indices, starting from the village level up to country level every year. And these data are collated and collected on a monthly basis in Excel sheets. Now, being in IIT, you know that Excel sheets of 720 odd districts spread over 20 years, for example, is a headache for anybody to go through data. So, this kind of data analysis will be very, very tedious to do if you wanted to look at trends, you wanted to understand disease epidemiology, you wanted to look at where are the new outbreaks happening, you wanted to see where malaria incidence is coming down very rapidly, you wanted to see where malaria incidence is stable, for example. Going through Excel sheets over 20 years for 700 odd districts would be tedious. And this problem was highlighted to me when I joined the ICMR system. So what we did initially was to actually make, using our software package, we made a malaria dashboard, which is really very nice. I'll show you the features of this dashboard, which is published in the Lancet series last year. And so what this dashboard allows you to do is all the data over the past 20 years in all the Excel sheets has been now condensed into digital form. And it allows you to do, using code within the R programming language, using codes embedded in it, it allows you to do both analytical analysis, overview of the data, as well as a visual overview of the data, because it's linked to GIS. So I'll show you some examples of that. So this is the dashboard. Essentially, it covers country-wide, district-wise, and state-wise data. Depends what you're interested in, because as I said, the country is too huge. If you're interested in malaria, for example, in Uttarakhand, then you wouldn't be bothered about Telangana. So you can click, for example, the state of Uttarakhand, and then look at the incidence of malaria. So this can be done very easily now. Instead of going through literally thousands of Excel files, you just select the state here, you select specific district, you can select multiple districts as well, and you can select a malaria metric indices. In this case, this is API, or annual parasite incidence, which is the number of cases divided by the population times 1,000. So basically, API of 1 means 1 case per 1,000. So you can actually do that, and so you can, very trivially, in an instant, in a flash of an instant, you can actually get these graphs, and you can see what the trends of, in this case, this particular district bar data are over 2000 to 2020 or so. So this malaria dashboard actually makes it very, very easy and facile to go through malaria data and analyze it from an epidemiological context. You can also do other things with this. For example, you wanted to see all the districts of Assam, in this case, in the year 2007. So it will actually produce graphs of the districts with the highest, in this case, highest API to lowest API. So if you want a granular analysis of where within Assam, in this case, what the problematic areas and where you could actually feel that there was some element of control, which is here, where a huge problems here, you can actually do it in a trivial way. And you can do this year-wise, and you can actually compare year to year as well using this dashboard analysis. So if you want to do, for example, here, compare between districts on the basis of API, you can actually select specific districts, in this case, Karnataka districts. Here are the districts that we've selected: Bangalore, Bidar, Dardwar, etc. And each of them would be graphed in a different color. And you can see again here the trends on the basis of this analysis, you could conclude that epidemiologically, malaria was on the decline in these particular districts in the year 2000 to 2019. So, this kind of analysis actually makes it makes very valuable contributions to understanding disease epidemiology, which is really important for the national program to then take measures for controlling of malaria, or in cases where malaria is under control, to then perhaps reuse or reallocate those resources in our towards spaces. You can also, for example, ask for percentage change from 2018 to 19, 17 to 18 in this case. And you can see here, cases where you would get alarmed if the cases were actually going up between as you progress ahead in time, which would perhaps potentially indicate that there were outbreaks or an element of control wasn't happening in these particular districts. And in the end, you could also actually, all the analysis is GIS mapped. So this is a GIS map from 2002 to 2018. And you can see here what is color coded here in the heat map is the API, or annual parasite incidence. And you can see the red areas and the orange areas are slowly shrinking in the past in this particular window of, say, 16 to 17 years. And really the problematic areas are shown here again, as I had alerted to you, are in Chhattisgarh, Odisha, and Jharkhand, etc. Bihar, for some reason, has always been very low for malaria, and it's not because there is no malaria in Bihar. It's just because there is absolutely no surveillance in Bihar. So that's not a good story. In any case, we won't go there. So you can see the progress of malaria in this way, and GIS mapping also then allows you to immediately give a visual impression if you were to go ahead and try to re-establish some control measures in cases where what you would call this as recalcitrant or persistent malaria states. So much so good so far. You have oodles of data from the program. You are able to analyze them. So what really is the problem with malaria control? And herein starts the sort of the molecular aspect of my talk. The problems with malaria control are the following, really. I mean, there are 3 distinct measures by which malaria can be controlled. You have very good insecticides, you have very good surveillance, you have very good diagnostics, and then you have very good treatment. And the answer to the last is yes, we have very good treatment. However, as you can see here, I'm showing you just the example for one particular drug, chloroquine, which became famous or infamous in COVID. And you can see how chloroquine resistance actually spread very rapidly after it was administered in the public health system. I'll just give you an example here of, say, India. Chloroquine resistance started in 1957 in Southeast Asia. And it rapidly migrated towards the Burma border. And from Burma, it entered into northeastern states, the Seven Sister States, went into China. It went into India. India transported it, or via India, it went into the Middle East. From India, it went to Africa. It went to Africa to the extent that chloroquine was the main drug for P. falciparum in Africa. It lost its efficacy completely, and literally millions of deaths happened over the course of decades because chloroquine was not working anymore in Africa. So drug resistance is a really major issue, not only for malaria, for other people who are working here in AMR, they will know this very well. But really, malaria presents the most classical example of how drug resistance manifests so rapidly. So chloroquine was introduced in 1945, and by 57, there were reports of resistance to it. Sulfadoxine, pyrimethamine are two drugs that target the folate pathway. They target two different enzymes, and I'm going to show you in detail about them. The same year the drugs were introduced, there was a report of resistance. Mefloquine within a few years, and artemisinin, which is the drug that Professor Yu discovered, has been noted to show artemisinin tolerance of failure within 7 to 8 years of its introduction. However, things aren't that bad for artemisinin as yet. Surprisingly, a lot of these drug resistance reports have originated always from the Southeast Asian part of the world, essentially from Cambodia, Thai borders, and it remains unclear what parasites are undergoing in this particular region of the world that allows them to rapidly generate drug resistance. And that's a real biological problem that remains unsolved. So I'll give you an example of the sulfadoxine pyrimethamine, how sulfadoxine pyrimethamine resistance manifested itself. Here's P. falciparum, which is one of the malaria parasites. Here's P. vivax, which is the other dominant malaria parasite in India. In India, you basically have a 50-50 distribution of the two parasites, although they are quite different in the way they manifest themselves. And you can see just rapidly after the introduction of pyrimethamine, you have resistance. After the introduction of sulfadoxine pyrimethamine combination, you have it as the first line, and you have immediately a report of sulfadoxine pyrimethamine resistance. And as you go along in time towards the right, from 1950s to 2000, what happens is that these genes that are actually gene products, the proteins that are targeted by these drugs, they start to show specific mutations in them, specific SNPs in them that counters the effects of these drugs, okay? And that's how drug resistance manifests itself in a classical way. That's not always the case. Drug resistance mechanisms and drug action mechanisms don't necessarily overlap, but we will discuss that later. So what happens is the genes that are being targeted, or the proteins that are being targeted, they start to develop single, double, triple, quadruple, quintuple, sextuple mutations in them. And every step of a mutation actually decreases the efficacy of the drug. Okay, so that's shown here from the same paper. So the target of pyrimethamine is DHFR, and the target of sulfadoxine drugs is this particular enzyme called THPS. And what's shown here are specific mutations shown in red that starts to occur in these particular proteins as a function of repeated administration of sulfadoxine and pyrimethamine. And these mutations, for example, this is quintuple and sextuple mutations, these are super resistant. So essentially, there's no point giving SP drug to somebody whose parasite genotype shows you these specific sets of mutations. Now, the question is, epidemiologically, what is the distribution of these mutations before we start giving drugs to people? So if you look here, what we had done was to do this analysis. This is wild type, single mutation, double, etc., till quadruple, in this case, red. So you can look at the distribution of South America, etc. But if you look at India here, for example, you can see there's already quadruple mutations, substantial fraction of them, and a substantial fraction of them are double mutations. So the drug is already compromised in India. Similarly, here you can see again, this is different for PF and PV. The drug, to a certain extent, in this case, this is P. falciparum DHFR. The drug is already compromised severely in some cases in India. And this was shown about 10 to 12 years ago. And the tools that you use for this are what's called genomic epidemiology, and I'll show you how we are doing that. So let's look at, because this is a department very, sort of very keen on structural biology, and I'm originally a structural biologist, as is my host here, Dr. Praveen Kumar. I want to show you how structural biology allowed us to understand what these mutations do. So I'll just pick one example here of one of these two enzymes where these mutations manifest. You can see a single mutation here, serine to asparagine, etc. All these mutations actually accumulate where the drug binds. Okay, but remember, the drug is actually a mimic of the substrate. So wherever the drug binds, the substrate has to bind at the same place. And since these enzymes are essential, you can't have a situation where the enzyme doesn't recognize its own substrate because the product is essential for folate biosynthesis in parasites. So in these quadruple mutations, although these mutations surround the active site, what all they do is they widen the space for the drug to bind. So the drug binds weakly, but the substrate still manages to bind very tightly. So how do you assess that? So what we did was we did this analysis where you actually measured the, I'm sorry, you can't see this very well in the y-axis label, but this is Ki divided by Km. So Ki is the inhibitory constant, right, of any particular drug, and Km would be the constant for binding affinity of the substrate. So what you can see in blue is the substrate affinity actually doesn't change substantially most of the time for any of the mutations. So the enzyme continues to recognize the substrate, but the Ki value or the inhibitory constant of the drug goes very rapidly. This is about 800. So this quadruple mutation here is 800 times worse than the wild type enzyme. So the drug is 800 times less effective than it would have been in the case of wild type. And this happens for both. Both sets of enzymes. Again, you see blue. It recognizes the substrate, doesn't recognize the drug very well, and that's how this drug resistance in this particular case is manifest. This is quite serious, and it was so serious that India had to wake up on this issue and look into this aspect because sulfadoxine pyrimethylin resistance had spread through most of the country at the time, 10 to 12 years ago. And so well, here's a paper that you can read on this issue. And so what it forced the national government to do, national program to do, they recommended to the government, and it subsequently happened, was that because of genomic surveillance, they noticed that in this Northeast, sulfadoxine pyrimethylin resistance was the maximal. Whereas in the rest of the country, it was subpar. It wasn't that bad for the moment. So in fact, India is one of the only countries that has two different drugs for malaria. I mean, as in the national government recommends two different drugs for malaria within the same country. So Northeastern people, if you were to get malaria in Tripura, you'd be given a different drug. You'd be given artemisinin lumefantrine because that works, and that's not SP. And the rest of the country, you get SP. So if you got it in, for example, Jharkhand, you will be taking SP. Now, this is a complicated situation itself because these people going back and forth do not need any visa. There is no checking on the borders. There is no borders within India. And so you can imagine a person traveling from the Northeast, which happens all the time, a lot of labor and education students come, etc., for business people come. They travel to the mainland. They're potentially bringing sulfadoxine pyrimethylin parasites to the mainland. They transfer it to somebody in the mainland, and that person is then given the same drug, ASSP, which is already compromised, and vice versa. So this isn't really the ideal policy, but I just wanted to home in the point how serious this is when you start to see drugs fail, and how a country as big as India would actually struggle with handling such a situation as is presently shown to you. So how do you do this genomic surveillance? Well, we've actually started this using genomic tools recently. And at the time it was tedious, but what we've done recently is, and I'll show you very briefly some data on this. So this is genomic epidemiology. So we actually collected DBS samples. So we basically pinprick positive malaria patients, that is in people in whom malaria parasite is evident by diagnostic means, and make dried blood samples, DBS samples. And then so these are, malaria positive cases, we collect their DBSs, etc., extract the DNA, and then we've established a multiplexing PCR technique, which actually sequences PCRs 15 genes simultaneously. Some of them are shown here, and some of these are actually drug resistance genes. For example, I already discussed the first two. The third one deals with artemisinin, and there are other drug resistance markers here. For example, CRT, which is for chloroquine. And then we do amplicon sequencing using nanopore. And the kind of readout you get is very interesting. So it's a busy graph, you don't need to worry about it. But if you just look at this particular one, DHFR and DHPS, so these are the two genes we've been discussing where quintuple mutations and sextuple mutations are important. From the y-axis is actually 22 individual patients from India. And wherever it is a red is a mutation. So you can see, for example, this particular person has 2 mutations, but so-called barcode 01, but you can actually go down and find people with 4 mutations, and there are even people with 5 mutations somewhere here. So, well, here 1, 2, 3, 4, 5. So this person has actually a quintuple mutation, which is essentially totally resistant to sulfadoxine pyrimethamine. And if he happens to be in the mainland of India, not in the northeastern states, then he'll be given the wrong therapy. And so you get a readout of the variants like this, and we've actually now extended this. We're literally doing thousands of DBS samples from across India to then have a genomic map which lists the variants so that to alert the national program about drug policies whether they're still relevant or not. So genomic epidemiology is where you need to be, I think, these days if you're involved in infectious disease. I'm going to switch gears and talk about what my lab has also been doing on the side, which is to try to look at some of these conserved enzymes within the malaria parasite as druggable targets. And I've, I mean, I'm sure all of you know very well that for protein synthesis, which is essential requirement in all cells, you actually need a set of 20 enzymes or so, which are essential in all life. And what these enzymes do is to put an amino acid with a particular enzyme together with the ATP initially, and then with the tRNA, it recognizes tRNA, and at the end, it actually ends up charging this, what's called a charging of the tRNA, so that this tRNA can then go to the ribosome for ribosomal protein synthesis. And this activity has to happen for each of the amino acids, depending on the mRNA readout in the ribosome. So that the tRNA has to be recognized as per the instructions of the mRNA. And actually, this is how genetic code is translated in all life. So these synthetases are very interesting. And we've been fixated with them for about 15 years now. And one of the things, there are several facets to them which I find interesting, which I feel are druggable, but I may be wrong. And one of the things is that they are very easy to assay. So, if you're involved in drug discovery, and especially if you're looking at some proteins or enzymes, it's really important that you have an assay system to be able to screen drugs or inhibitors. And so these enzymes are actually called Rs for tRNA synthetases. And so the first part of the assay actually releases pyrophosphate, which can be easily detected in this particular assay called malachite green assay. It's a colorimetric assay. And the second part of the reaction, if you give it tRNA, can also be easily detected because it releases a product called AMP, of course, and there are AMP detection kits. So that makes really very easy to detect both parts of the reaction. First, making of the amino acid adduct, and second, charging of the tRNA. So you can have a complete high throughput assay system for any particular enzyme using a colorimetric assay, both are colorimetric assays, and have a readout whether the enzymes, first of all, are functional or not, and secondly, whether they can be drugged or not. And so we started this exercise some 15 years ago, and I'm just going to recap for the malaria parasite. What's shown here in red is the infected RBC, which is where the malaria parasites reside in when they infect you or me. And all the malaria pathology is actually associated only and only with the red blood cell stages of the malaria parasite, as far as we know as of today. The malaria parasite is an epicomplexan, so it has actually 3 compartments. It has an extra epicoplast, which is sort of a hangover from evolution and endosymbiotic event where it ate up a bacteria. In any case, so epicoplast is translationally independent, and so the protein synthesis in this malaria parasite and in all epicomplexans except for crypto has to happen in the cytoplasm, in the mitochondria, and in the epicoplast. So you really have 3 sites of protein translation, right? And we discussed earlier that you need 20 enzymes. So you would have thought that the parasite actually encodes for 60 enzymes for each compartment, but that's not the case. It actually encodes only 36 enzymes. And we are not going to go into this in detail, but what we did a few years ago, was to make a periodic table of these 36 enzymes from P. falciparum. And what's shown here are the ones in sort of magenta are epicoplast bound. The ones in green are cytoplasmic, and these 4 are dual localized. So they have specific signal sequences that allow them to be resident both in the cytoplasm as well as in the epicoplast. And that's how you are able to get a complement of all 20 in each compartment. The mitochondria doesn't really encode any, all the charged tRNAs are migrate into mitochondria for mitochondrial protein translation. So the ones in the green are of specific interest because they are in the parasite cytoplasm, and they are the ones that we've been focusing on for drugging. So over many years, we've actually looked at several proof of concept studies for these enzymes, whether they can be drugged. And if you are interested in some enzyme system for drugging, what you need are drugs, or you need inhibitors, or you need some potent compounds that do something to the enzyme. Just solving the structure on its own, in the absence of these, is never going to lead to a drug. And so we picked these enzyme systems initially, um, and we were able to solve structures of several of them. I'm not going to talk about each of them, but I'll discuss one of them, which I think is rather more interesting than the other two, which is this phenylalanine tRNA synthetase, whose structure is shown here. It's a heterodimeric structure of two subunits, alpha and beta, which forms a tetramer, in fact. So it's rather one of the largest tRNA synthetase assembly that we know of. So the phenylalanine tRNA synthetase is very interesting for many reasons. My student Arvind, many years ago, at the time we weren't doing structural biology of this enzyme, we were interested in why P. falciparum, it has 3 genes for phenylalanine tRNA synthetase. Well, it has an extra gene. So if you look at the distribution of these FRSs amongst epicomplexans, and I told you that malaria, toxoplasma, crypto, etc., are all epicomplexans. Some of the other ones are listed here also, like the BCA, etc. You can see that P. falciparum has 4 particular genes for FRS, for phenylalanine tRNA synthetase. Because it forms an alpha-beta heterodimer, there are actually 3 protein assemblies. But this is unique to P. falciparum. If you compare this to, for example, toxo, you see absent. And if you compare it to crypto, you see absence of 2 of them. So there was something special in P. falciparum about phenylalanine tRNA synthetase that wasn't the case in other epicomplexans, although they are fairly related parasites evolutionarily, not in terms of the diseases they cause. And so we had actually started working on them. And at the time, it was this group from Stuart Schreiber's group in Broad Institute got in touch with us because they had been doing some very clever chemistry. Now, Stuart Schreiber, I don't know if you know of him or not, but you should go to the website and check him out. I mean, I think this guy has some 400 patents on drug discovery, and this is what he does. He does clever chemistry for drugging of enzyme systems. So what they did was they actually created synthetic compounds that mimic natural compounds. Now, natural compounds are not only natural because they come from nature, but the specific stereochemical physical characteristics they have is that they have a very set 3-dimensional structure, which usually synthetic compounds don't have. I mean, they have a 3-dimensional structure, but it's not very 3D. So they synthesized some 100,000 molecules, and they did this mass screening against the malaria parasite. And what they discovered was that one particular compound, which is shown here, was able to kill all blood stages of the malaria parasite. It was also able to kill all transmission stages of malaria parasites. It was also able to do prophylaxis in an animal model system, which would be that you take the drug and then can't get infected by the malaria parasite. And it and that shows that likelihood of it having an enzyme as a substrate, or rather as a receptor, because there is a 3-dimensionality involved into the recognition of this stereochemical compound. So you can see here the potency of this particular compound is so low. It's in micromolars, 0.009, and people who work on AMR and other infectious diseases would know this would translate to nanomolar EC50s, which is really amazing. So it looked really good, and we got involved in this project at the time. And the first thing we did was to ask the question whether this compound in fact interacted with phenylalanine synthetases. And we had some other synthetases as control. And what we could show was that yes, indeed, the IC50 is the inhibitory constant for the particular drug. With purified enzyme, EC50 is the effect of the drug on the cells. And if the two of them correlate like this with very good linearity in this between the two, very good correlation between the two. This essentially implies that the drug binds to one enzyme and kills the cells with specificity. Okay, and then what we did was actually a series of mutations were generated artificially in the parasites, and what we were able to show was that the enzyme, the native enzyme, of course, kills the parasites very well. But as you have mutations in this particular enzyme, actually the mutations are in the gene, and they manifest themselves in an enzyme which is not able to bind the drug very well, exactly like the ST story I told you earlier. You start to shift the EC50 to the right, so it becomes poorer and poorer. That again defines that there is a specific target, and this is likely the target. We were able to then map the mutations, although this was just a homology model, it was probably not correct at the time, but they did sort of fall within the space of one protein unit. And specifically, the most potent compound, the most potent compound was used for doing assays for IC50, and we could show a significant shift of just one mutation that causes a significant shift in the enzyme inhibitory. So that again implies that there is very specific interaction between the two. I'll labor on about this point because specificity of interactions is really important when you're discovering drugs against pathogens, because you obviously don't want to kill the host. And so you can see here, when you calculate KD value for this enzyme inhibitor complex, the KD value is 7 nanomolar for the parasite enzyme. Please don't forget that the same enzyme exists in humans, because all cells have to have the same enzymes. Even bacteria, anybody will have the same. The exact corresponding enzyme has 1,000-fold worse KD value. So there is something in the structure of the human enzyme that is unable to recognize the drug as well as the parasite enzyme does. And this is what you want. The selectivity index in this would be this divided by this, which would be about 1,000, which is very good for discovering inhibitors. I told you the drug is, the enzyme is fairly complicated. It is a large protein of alpha beta heterodimer. We were able to solve the structure, and here's the drug. And what's peculiar about the drug is that it has this diaryl acetylene moiety. It has this cyclopropoxy phenylurea moiety. The urea is here. This cyclopropoxy is very peculiar, and it was all based. All these compounds were based on chemistry that had in common bicyclic azitatine as the scaffold, as the main scaffold, and these were both extensions. And you will see what I mean when I say that this mimics a natural compound, because what you see here is just two-dimensional, but you'll see the three-dimensional structure is rather striking. So we were able to solve the structure of this FRS enzyme with this particular BRD drug. And so you just bear with me. We're talking about this particular compound. I'm sorry about this. And so in the enzyme active site, this is where the substrate of the enzyme will bind, which is L-phenylalanine, the amino acid. This is where the adduct will be formed between the phenylalanine and ATP. This would kick out pyrophosphate, right? We discussed this. And the tRNA will come from this side. And here's the drug. We actually solve the structure from both P. falciparum and P. virax. And the drug takes a very sharp turn. It's actually a very L-shaped drug. You can see here, the cyclopropoxy moiety goes in this particular pocket. And the base of this, the azitatine, is at the center. So it's actually a right-hand turn. And the biphenyl diaryl acetylene moiety goes into the L-Phe pocket. So now the mystery is somewhat resolved, because the way this compound works is this particular ring mimics L-Phe. It mimics the substrate, and so it's a substrate substrate mimic. Only this ring, this particular ring bicyclic azitatine doesn't recognize anything, does not go to the ATP site, and it's just hanging around between two sites. And the cyclopropoxy goes into a new subsite, or what we call an auxiliary site, which has no known function. And that's important. So that is the mechanism of action of this drug. It works by The RNA would go here, amino acid goes here, auxiliary site here, and ATP site is here. And our drug, particular drug, goes in this fashion. It's L-shaped drug. The propoxy unit goes here, and the biphenyl ring goes here, mimicking the L-phi. ATP site is empty, and the tRNA site is also empty. So that is the mechanism of action of this drug, and it's really remarkable, but it seems to work. So anyway, this was published. We published both the PF, our work on the P falciparum enzyme as well as the P vivax enzyme, and um, and then we observed other phenomena which was really interesting, and this sort of kickstarts the next phase of my talk, away from this particular story. Um, if you do phylogenetics, you'll see that P. falciparum, vivax, nosei, this is also a malaria parasite and it does infect humans, are of course related to crypto, etc., and toxo and leishmania and malaria well, because many of them are epicomplexins, leishmania isn't. And if you were to look at EC50 values, the drug is very potent here, very potent here. We did against nosei, potent here, against crypto as well, and against toxoplasma as well. So what is the reason that the drug seems to work against so many pathogens? And the answer for structural biologists would be trivial, which would be that the active site is highly conserved. And in fact, if you look at the active sites of these other enzymes, I mean, of the other FRSs from other parasites, you'll see, and if you were to do a lineup of the residues, you'll see that there's high conservation between toxoplasma, PV, and PF. And there is something distinct about the human enzyme, and that's exactly why it doesn't really recognize the human enzyme. In fact, the human enzyme is not recognized because the auxiliary site residues are somewhat different. So this means that you can actually have a drug or an inhibitor that can target multiple pathogens that are related somehow, as long as there is conservation in them. And you don't have, if you don't believe me, I mean, both studies are published in, this is against crypto FRS and this is against the toxo FRS. So this actually works very well. So we gave this a name of what's called STOP, right? And we came up with this idea of structure-based targeting of orthologous pathogen proteins. And I mean, for structural biologists in the audience, this would be trivial, but for others, it's actually a very facile way to think about it. You have an enzyme structure with a drug bound to it. You do multiple sequence alignments. You make a list of the residues that specifically interact with that drug. And then you screen the database for all other enzymes that exist where those residues are conserved, because then you have a hypothesis that if the residues are conserved, the same enzymes can be easily targeted by the same compounds. And so that's the bit about structure-based targeting of orthologous pathogen proteins. So they're they're sort of completely different organisms, but by virtue of conservation in the active site, they may be targetable. So the question is, does it work? Well, somewhat, yes. We have examples from our lab and other labs where this thing works for leishmania, toxo, crypto, malaria, etc. Cladosporin, I didn't talk about some of these other compounds. One is from our lab and two are from other labs, where they targeted lysine tRNA synthetase, and a lot of work from multiple labs have shown that Loa loa parasite and Schistosoma mansoni, which are both eukaryotic parasites, the helminths like, can also be targeted by the same compound. In this case, this particular series of compounds targets leucine tRNA synthetase, and it can target not only malaria, but candida and toxo as well. And in this case, this particular compound series targets the proteasome in completely unrelated parasites from malaria, Donovani and Trypanosoma brucei and cruzi. So you have this sort of cross-species targetability using a compound as long as it's targeting a conserved enzyme. So we just recently wrote we were asked to write a review on these tRNA synthetases in pathogens. And so you can see here, we've actually summarized this in terms of the context as current knowledge of today, which are these particular pathogens here, Brugai, Cryptosporidium, Giardia, etc., Leishmania, etc., and here the trypanosomas. And the structural information on these systems is really very sparse. And if I was starting my career over, I would start working on all of them. Because I think a lot of them would be targetable by the same compounds that we and others have discovered, just by virtue of extension and extrapolation of what I've shown you. So there's a lot of, I don't know, another lifetime of work that can be done on these, I think. And many of these happen to be neglected tropical diseases, so that makes it even more important. But from a philosophical point of view, okay, fine, tRNA synthetases can be drugged. nothing, nothing that surprising about this, I think, as long as you can discover a potent inhibitor. But intellectually, what's more challenging is to answer these questions. Can two drugs bind to the same tRNA synthetase? In fact, the question whether two drugs can bind to the same enzyme at all. And I don't mean two inhibitors, I don't mean two substrates, I mean two different drugs. And a related question, can two tRNA synthetases be targeted by the same drug? That also is a very potent idea. Here, you have the conserved target and you are hitting it with drug A and drug B at the same time. And here you have the same drug A hitting two different enzymes. So people who are savvy with some of the stuff in terms of genomic evolution would know immediately that both have their implications. In this case, drug resistance would be far harder to get because it will have to mutate a lot more residues. And in this case, again, drug resistance would be harder because the parasite in this case, or bacteria, whatever pathogen you're interested in, would have to generate simultaneous mutations in two different genes. So does this work? Well, just to summarize quickly again, so far in the malaria parasite, these are the potent inhibitors that we and others have discovered. Halofignon, cladosporin, and azacitidine were done in our lab, and bernardine and benzo compounds were discovered in other labs. The summary is that all of them go to the active sites of tRNA synthetases. The question is whether more than one can go to the same active site. So we hypothesized whether more than one can go to the active site many years ago, because all we did was there were structures available with independently of substrates and of independent inhibitors. So we just overlaid them and asked the question whether the inhibitors and substrates overlap with each other. And we discovered that on the whole, they don't overlap with each other. So the active sites are actually big enough and large enough and plastic enough that they can accommodate multiple molecules. And so we were actually recently able to prove this, as it's just published, in the case of a tRNA synthetase. So here we show two different drugs, which, by the way, independently are highly potent on their own accord. Halofignon and ATP inhibitor called L95. It mimics ATP. And here's the density, electron density for L95. It's bound in the presence of L-proline. This is an independent structure. And here's L95 bound in the presence of HFG. So both of them are independent drugs, very potent on their own. And they are bound to the same enzyme in the active site, and they really barely contact each other, as you can see here. L95 with L-proline, ANP with halofignon on its own, and here's the double drug complex of L95 and HFG. And this, to my knowledge, is unprecedented because this opens up the avenue to target these enzymes with multiple drugs at the same time. Now, can you extend this argument a little bit more in context of malaria? Because it's okay to kill the parasite, but I, at the very outset, said, and you already know that malaria is actually spread by mosquitoes. And if you could contain mosquitoes, I mean, mosquitoes don't only spread malaria, they spread a lot of other diseases. And so, containing mosquitoes would be one way to stop transmission. And you have that thought in your mind, and you have a second thought in your mind that as long as enzymes are conserved, they can be targeted by the same compound. So we naturally made this extrapolation to ask whether the same enzyme in the mosquito can be targeted by the same drugs. And so we did this experiment with larvae of Anopheles culicifaces. These were exposed to halofignon, the drug that I discussed in the previous slide. And you can see in untreated larvae, and as per treatment with higher doses of this particular drug, you can see huge disfiguration in the midgut. And we've actually now done histology of these treated larvae, and they have significant amounts of inflammation in the midgut. In summary, they don't last for long and they die. And so we were able to also show that the corresponding Anopheles culicifaces enzyme is actually targeted by halofignon. We calculated its IC50. And we also now have a 3D structure of the two drugs bound to the mosquito enzyme. In this case, you can see with the electron density for them. And so, but more importantly, in assays, we were able to show that so here's percent mortality of the larvicidal activity of HF culicifaces. And this is a related mosquito vector in India, Stephen's eye. And you can see the mortality is quite significant. At 50 micromolar, at 100 micromolar is almost complete. And similarly here at this high concentration. Now, this doesn't automatically make them larvicides. This is just the first step, but there's a whole potential for them to be larvicides. Remember that any toxicity associated with these inhibitors may not be necessarily manifest and become a concern because humans don't naturally, humans are not going to be drinking the water where mosquitoes breed, I hope. So, this idea of targeting the same set of enzymes in multiple species and multiple vectors possibly can work. So I'm going to, see, I might have taken too much time, but I'm going to come back to malaria epidemiology and why I mentioned to you that malaria can be used as a biomarker for national development. You really need a huge number of agencies and stakeholders to be able to control malaria. You need these agencies to work in synchrony. You need these agencies to have sufficient funding, a lot of crosstalk. You need these agencies to be modern, to be sharp with the latest tools of molecular science. And specifically, you need these agencies to actually be able to perform what they've been able to do, which is, be compliant to whatever the public health policies are of the case. All this kind of synchronization is non-trivial, and it becomes even more non-trivial in countries where, which are very large and which are very heterogeneous, which is the example for India. And so I'll start with well, not start, but I'll just give you one example of Sri Lanka. Sri Lanka eliminated malaria many years ago. And people who are in malaria always give the example of Sri Lanka as, well, if Sri Lanka can eliminate, why can't we? Well, Sri Lanka is, I mean, I hope there are no Sri Lankans in the audience, but it's I would say it's more homogeneous than India is, of course, notwithstanding that it's much smaller. But Sri Lanka, had a hard time controlling malaria. If only the lessons of Sri Lanka can be transplanted in India, I think also that would be very beneficial. So I'm showing you the cases of malaria in Sri Lanka, and you can see here the peaks from 1911, say, till 1950. And so it was very significant problem there. And in 1950s, basically, malaria was essentially eliminated from Sri Lanka. And what happened at that time was the public health and the government and the bureaucrats and technocrats all had a cigar and said, well, we've controlled malaria, it's end of story. Because the caseload was very low for many years to come, even for a decade to come. So they were essentially celebrating, as one should. But malaria then resurged. And then it came back and it stayed on for another 30, 40 years. So malaria is not for nothing the oldest disease that has existed on Earth. The moment you take your foot off the pedal of malaria control, it will resurge. And so if you've been following literature, and if not, I'll tell you, WHO has already alarmed in the past 2 years that because of COVID, the implementation of malaria control across the world suffered, rightly so, and because the public health agencies were busy containing COVID. And so there is already what's considered a rebound of malaria happening. Actually, malaria incidence is going up in many bits of Africa and in some parts of India as well. And so malaria is a very tricky disease. And so, you will see this kind of a resurgence the moment you start to look the other way or pat yourself that, well, we've conquered it, till it's not conquered. And of course, after this, they remained, Sri Lanka in this case remained malaria free for several years, and then they applied to WHO for a certificate of elimination. As you know, Sri Lanka is malaria free, so is China. And you need to show evidence that there is no indigenous transmission of malaria for 3 years, and then the WHO teams come, they verify your reports, and then only you get a certification. So it's not easy to actually eliminate malaria. It wasn't easy for Sri Lanka, and it's not going to be easy for anybody involved in this business. But what we can do is the following, and I'm just going to end there. Is my hero, Charles Darwin. I'm sure you recognize him. And it says, if the misery of our poor be caused not by the laws of nature, but by our institutions, then great is our sin. And so, the misery of the poor, in this case, victims of malaria or any other infectious disease, this is a natural phenomenon that happens on Earth because there is a struggle for survival between pathogens and ourselves. But humans have built institutions to counter that. And if those institutions are not performing at the level that they're supposed to do, then it's only our sin. We can't really keep blaming nature for it. And I'll stop there. My last slide would just be thanking the people really profusely who've been working with me for many, many years. These are some of the publications that we've been associated with, and show you a photograph of the people that I worked with, which has always been a delight. Thank you very much for your attention.
Thank you very much, Dr. Amit, for a wonderful talk. Although this is a memorial lecture, we are not supposed to take questions, but if you allow, can we have
Yeah, sure.
a few questions? So I think from audience.
This talk. So, Stuart Schreiber's molecule. So, it's kind of interested in your line of thinking as to why would it target TRN phrases, or since you had the enzymes, you said that they were stereospecific, so that's why you thought that they would be. But there are so many enzymes, right? So why did you think of only targeting TRN synthesis, or is it because your lab had access?
Well, that's a good question. So, of the 100,000 compounds that they screened, they actually discovered a few hundred that are potent killers of malaria parasites. And in almost all cases, they didn't really know the target, right? Identifying the target is very difficult. So they had started to do selection of parasites to look for mutations that switch the IC50. Yeah. And so they had some evidence initially that one of the genes that was targeted was the alpha subunit of the tRNA synthetase. And so they were in touch with the Medicines for Malaria Venture, which is this funding agency in Switzerland, and that agency funds our lab. So they put us in contact with them that this is the tRNA synthetase lab, and maybe you should verify what you're observing now. So unless you have evidence, genetic evidence or otherwise, that the target is that particular gene, then it will be very tedious to go through thousands of genes to identify the target. Yeah.
It's very nice to know that on many, because on the same lines, like it's a different different kind of, but they're different substrates. And it will be, kind of, I would say, miraculous to develop, kind of some compound drug, which probably can target many set of, so, whether your wrestling was to kind of find out some conservation on the active side property, which should be targeted to develop a drug, or because to develop a drug for some reason which might be kind of changing for different amino acids. What was your rationale of developing maybe a drug, maybe a pocket of two drugs or three drugs, which can target a number of amino acids?
Yeah, I mean, so actually, I haven't shown you some recent data with this. So there are two aspects. One is that these inhibitors are tunable in the sense that if you were to recognize what makes for example, I showed you for FRS the diacetylene moiety that recognizes L-Phe. Now, in another world, you could modify that L-Phe mimicry to L-tyrosine, which is just an extra hydroxyl group, and you could switch the specificity of that particular inhibitor. We haven't done this for this, but we've actually done this exercise for a new battery of inhibitors. I haven't been through it, but I didn't show it to you. But yes, so I like the idea that some of these inhibitors are tunable as per the amino acid. Each synthetase has its own specific amino acid recognizers. So if you put it to a conserved pocket in these, for example, to the ATP site, and then you can tune the specificity via the amino acid. Having said that, we tried, and in some cases, it's very difficult because if you look at the ATP sites of tRNA synthetases, they are not conserved. So I had a postdoc, highly enthusiastic computational biology postdoc, and after a year he left. I mean, we had good terms and all, but he was given this project of recognizing the extent of conservation in ATP sites, never mind tRNA synthetases, in the whole of PDB. And it turns out, because of convergent evolution, I presume, that ATP is recognized by literally hundreds of solutions that have come up in terms of three-dimensional space. So there isn't one ATP site. Kinases have a slightly different one. Within synthetases, there's a diversity. So if you were to design a compound that goes to one ATP site, it will be very unlikely that it recognizes another one. I mean, and we know this from cancer therapy. I mean, kinase inhibitors fairly, SARC inhibitors are fairly specific for the ATP site. So because there is a huge plethora of recognitions available to nature to recognize an essential molecule like ATP, it has come up with solutions. So to engineer something that is cross-reactive is, I think, going to be quite difficult. You can just use it, AMP. So actually, most recently, what has been done is instead of AMP, now an AMS compound has been made, and actually for decades people have known about it, where this sulfates instead of phosphates. And AMS is highly non-specific. It goes and sits in many active sites and kills the bug. Now, therein the problem is how to generate specificity. So yeah, I mean it's a little complicated, but yeah.
Just I think because I mean, like what you said, for developing vaccine for the malaria parasites. Any effort I recommend some efforts have been made. What is the kind of what are possibly the status of getting vaccine?
Yeah, yeah. Thank you. That's a really good question. I always don't discuss that, but I should be doing that. So WHO has approved one vaccine, which is called RTSS, which is based on a circumsporozoite protein, and it is approved only in children from 0 to 5, if I'm not mistaken, and only approved in countries which have very high or medium levels of transmission. Now, it so happens India is very low transmission category by 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 the benchmark of Africa, where it's much higher. Um, However, the efficacy of RTSs is quite poor. It's about 35%, and children have to be given 4 doses, if I'm not mistaken, or even more doses to get that level of efficacy. Now, you can imagine, I mean, let's not discuss Africa. You can imagine a state like Jharkhand or Chhattisgarh or Bihar or I don't know, whichever state you want. Uttarakhand would struggle with that kind of thing. Repeated dosing of children for a malaria vaccine where the efficacy is 35%. I don't think the public health system is ready for this kind of investment. There is a new generation of the same vaccine that's been made called R21, which claims to have efficacy of 70%, but in fact, in real world, it may or may not have. I'm not sure. The data are still being analyzed. If it's 70%, fine. But both vaccines are targeted against P. falciparum. Now, India is in a peculiar situation where India has both P. falciparum and P. vivax 50-50. So if I was to pick a district, I don't know, let's say Dehradun district, I belong to Dehradun anyway. So let's say Dehradun district, and I said, okay, let's give P. falciparum vaccine to everybody. But the next day, the kid might come down with PYVAX. Now, you can imagine the fight back from the community that, oh, they thought they were protected by malaria, but in fact, they've come down with malaria. For them, it's all the same. They get fever, they get nausea, blah, blah, blah. So India is a peculiar situation where deployment of vaccine for any one pathogen, one of the two will be difficult. So I think it's very early days for India to think about that. Yeah.
Just to ask a quick question. Yeah, wonderful talk. So my question is, the agitation compound you have shown, which have a 4-member and a 3-member ring, which are friends. So how stable the molecule is?
The BRD compounds are highly stable, metabolically stable. We've done all the studies. And actually, that's a really good question because the drug people are looking for is in malaria should be single dose, and it should have very long half-life.
Yeah.
Okay, because artemisinin, the classical one for which Professor you won the Nobel Prize, is extremely potent, but it has a very short half-life. Within an hour or so, or 1.5 hours, it's metabolized. And so it's very good to bring parasitemia down sharply, but after that, parasitemia persists, which is why there is no artemisinin monotherapy. Artemisinin is always given with another drug. The second drug always has a longer half-life, so it persists to kill the rest of the remaining parasites. So this one has a long half-life, and that's evidence that is given there.
My second question is, as a drug designer, what is your philosophy? Because you have shown 2 molecules binding to one plate. So is it like combinatorial targeting one enzyme with 2 drugs, or taking folate biosynthesis and this one and targeting as a
So my rider there is that I'm not a drug designer. I'm a structural biologist with very humble origins, just like Dr. Praveen Kumar. But we've been lucky to work with very good chemists. And there are 2 pathways going forward for the double drug. One is to just do double drugging in a animal model system, because what happens is that if the drugs synergize, Yeah, if you see synergy in them, then the EC50 value will come down. For example, if you're giving 10 micromolar of each, if they're synergized, then you'll only have to give 1 micromolar or something because they have a additive effect in this case. If so, that's one avenue. The second avenue would be to persuade some chemists, like we've done. They're difficult to persuade sometimes, but to combine the two drugs and do some new chemistry to link them up, and to then do new chemistry to generate one molecule with peculiarities of both. But then you have to be aware of this Lipinski's rule, etc. It shouldn't become too big, over 500 daltons, etc. Hydrogen bond. So yeah, I think it's going to be a slow march forward. But we were very keen to just show that it's possible to do. Yeah.
Thank you so much, sir, for wonderful. I had two questions. One question was asked from sir, and my second question was, sir, there are many incidents of co-infection, like malaria with SARS-CoV and malaria with typhoid. So, can that anti-parasitic drug be used as antiviral drug also on the same time? Because in the era of SARS-CoV-2, we have seen that SARS-CoV-2 patient also suffering from malaria.
Yeah, that's true. That's true. But I mean, see, based on molecular evidence, nothing suggests that this drug should work against virus, because for that the virus will have to have a tRNA synthetase. And to my knowledge, most of the infectious viruses don't have tRNA synthetase because they use the host machinery for it, right? But if you were talking about a bacterial system, for example, Salmonella or something like that, then they will have tRNA synthetases. But getting this drug through malarial, through bacterial cell walls and all is not going to be trivial. So I think there will be specificity, to the extent that a double infection may not be curable. However, because of the conservation of the target. So that might work where there is a structural underpinning of why it should work. But randomly speaking, any other like MTB plus malaria, I don't see why that should work. Yeah.
And the other question, as you shown in your presentation, that two drugs are altogether binding at active site altogether. So, is there any experimental validation of synergistic effect of those compounds?
So we looked at it's in the paper. We looked at synergy, and there is very marginal synergy between these two drugs. Yeah. So to look at synergy, you need to make these isobolograms and so on. We've done that, and it doesn't look. This is very early days. I mean, one of the compounds has to be tweaked, and the synergy may come through. But these particular compounds don't show strong synergy.
When you were saying toxoplasma gondii, I think,
Yeah.
and then how about this Leishmania donovani? It must be having similar sort of structure in Leishmania donovani also.
Yes, yes. So we are actually. I mean, I just tell you. I mean, I had gone to Patna to a hospital, and I saw these people who suffer from Kala Azar. Actually, these are parasite-free people, but they get this dermal lesions, which are really terrible, and also filaria patients. This. This elephant leg and so on. I mean, so I came back and we've actually kickstarted this thing to look, because they are both neglected in a way. And we haven't done the experiment, but we are hoping that the same thing, if the enzymes are conserved, it should work. This is one of the issues. Malaria, tuberculosis, and TB we know, and so on. Even
when
some.
you were showing that one of the figure that toxoplasma and then Leishmania donovani were very close.
Yeah.
So
phylogenetically, they would be. I mean, the kinetoblast, they would have. Of course, the FRS would be conserved. We've looked at the active sites, and it's suggestive. Yeah, absolutely. And which is why I think this this thing of targeting orthologous pathogen proteins could work.
any other student in the audience? Thank you. Then let's give a big thank you to Dr.
Now our Dean Alumni Affairs, Dr. Patharo, and Dr. Ashwini, Dean Faculty Affairs, is Director Representative, to give a token of memento to Dr. Sharma. Please come over here. Both of you can. It's very heavy, so we need, and they both are very, weightless, weightless people.
It's very happy.
Yeah.
My hand is already injured, so.
You can use the other hand.
You can just touch it.
In the way, right?
You can just.
It's a very nice talk. Very nice.
So thank you all. So I think you had already tea, coffee before this, and let's go for lunch. Thank you so much. Yeah. And students or faculty members, if they want to interact with Dr. Amit, so let me know, and I will ask him if he has time to interact with you guys. But you can send me a message if you are interested. Okay, thank you.